All right, so we're going to cover in this chapter some new things. So in 16, we're going to add in a whole new category of things. Though we've talked a little bit about prokaryotes before, we're going to continue to talk about those. We also will talk just briefly about protists. Actually, we'll spend a whole lecture on it, but we probably won't get to it entirely today. If we talk about it, it is brief today, because that'll be the subject of our next lecture. So prokaryotes. So our essential question, the thing we'll attempt to answer today, is what are prokaryotes, which we know a little bit about. But we also want to classify them. So, and how are they classified? I'm going to apologize now because there is a ton of material to this section. So really it's important to focus and pay attention to what parts we need to pay attention and write down notes on and which ones we don't, because there is a lot of stuff here. Okay, so prokaryotes are the smaller of the cells. So these guys are smaller than eukaryotes. They can be about 1 to 5 micrometers in diameter. Eukaryotes usually range from 10 to 100 micrometers. So they're tiny. The collective biomass of prokaryotes is at least 10 times that of all eukaryotes. So although they are tiny, they're absolutely abundant. There's just a ton of them. Okay. So here's an example of kind of the tip of a pin, and was just the amount of bacteria that's there. And then we're talking about like a little push pin. So prokaryotes, again, just everywhere. And they're really diverse. So they live in habitats that are too cold, too hot, too salty, too acidic, too alkaline or basic, that eukaryotes normally couldn't survive. Some bacteria are what we call pathogens, and that means that they cause disease. Most bacteria, on the other hand, are beneficial or benign. They don't cause any problems. And many of them are on our bodies. So we have a thing called microbiota. You don't necessarily have to write this part, but it's at least interesting to know, particularly as it's relevant to things like, what do they say, like the probiotics and stuff. So we have a microbiota, which is a little kind of life thing, little community of microorganisms that live in or on our bodies. So each of us harbors hundreds of different species and genetic strains of prokaryotes, including a few whose positive effects are well studied. So again, those kind of intestinal bacteria that have essential vitamins, and we can extract nutrition from food molecules. So those probiotics is what they call them. And we have many bacteria that live on our skin and perform helpful housekeeping functions like decomposing dead skin cells. Okay. Handy little dudes. Other prokaryotes guard against pathogenic intruders. You're their body and they will protect you. So you gotta love these little guys because they will protect you. If something harmful comes around, they're like, no, this is my human space. So we like them. All right, prokaryotes in the soil actually help us decompose dead organisms and other organic waste material, which return vital chemical elements to the environment. If we lost the prokaryotes, the whole kind of chemical cycles that sustain life would just stop. It'd be all done. All forms of eukaryotic life would be. The drama. You gotta have drama with it. There are two very different types of prokaryotes, which you do need to know. These are our different domains, so do remember it is domain and then kingdom, phylum, class, order, family, genus, species. And so we have different kind of domains, and these two are our domains. So up here. Oh goodness, I'm tired. All right, so prokaryotic cells have 3 common shapes that are normal to know. They're helpful. They're actually part of most names. So if you have, for instance, strep throat, that's called streptococcus. And so the cocci is a spherical shaped prokaryote. Okay, so as there's lots of kind of, I don't know, circle shaped letters, cocci seems to work okay, I think. Bacilli, you can think of these kind of rod-shaped letters, are prokaryotes that are rod-shaped. So usually this is kind of our typical bacteria that we think of. It's kind of this shape, maybe like pill-shaped. They can also be kind of thread-like, and that's mainly because you just attach a bunch of those together, and by golly, you've got yourself a thread. Okay? We do have these cool ones, which we call, they come, let's see if I can use words this evening, but they have some spiral-shaped ones. So they have a couple of different names. If it's short and rigid, it's spirilla. And if it's long and maybe more flexible, we call it a spirochete. Okay? So we'll talk about those. We'll look at some things. So examples, much better pictures that I could draw. Cocci are the circle shapes. Bacilli are the rod shaped. And we have spirillus, or this one's spirochete because it's very long and spirally. Okay. Nearly all prokaryotes have a cell wall. So that's a kind of distinguishing characteristic. It provides them with some physical protection, and it also helps them from bursting in hypotonic environments. So if you remember, a hypotonic environment, there's going to be more stuff within the actual cell, and there's maybe more water on the outside. Well, we know water is very, very friendly, and it likes to go towards the inside, so it might give a tendency of a bacteria to swell. But having that cell wall on the outside prevents busting. We can't go past the walls of that. Okay, just a reminder. If we can ever get to this, I would love to show you guys, but there is a way to do this. You can gram stain and a bacteria, and so it will test for peptidoglycan. So a gram-positive bacteria has peptidoglycan in the cell walls. Gram-negative have less of it. Okay, and these bacteria are usually more complex and usually cause disease. They're a bummer. Okay, so there's some gram staining where you can see the gram-positive ones are more the purpley guys, and these gram-negative ones are the little pink dudes. So the cell wall of many prokaryotes is covered by a capsule. It's kind of a sticky layer of polysaccharides or proteins. So the big thing to know is maybe that there is a capsule. Some basic anatomy of it. It enables the prokaryote to adhere to their substrate or to the individuals in a colony, and it also shields them from attacks from the host immune system. Because our immune system likes to fight things. All right, so there's a little capsule. Maybe this is a tonsil cell and this is a capsule of the bacteria. So prokaryote population growth occurs by binary fission, which is a lot it's like a simpler version of mitosis. Just to kind of review. Very simple and not exactly the same, but along the same line. And they can rapidly reproduce or produce new generations within hours. And that can generate a great deal of genetic variation by spontaneous mutations, and increasing the likelihood that some members of the population will survive changes in the environment. So for those of you guys who kind of said something about on the quiz that it takes a long time, it's not necessarily about that. It's these guys can do this kind of mutation and evolution stuff quickly. The genome of a prokaryote typically has about 1/10 as much of the DNA as a eukaryote genome, or 1/10, 1/1000th. There we go. So it's a lot simpler. You don't need a whole lot of genes there to power just one single cell. Okay. It's usually a long circular chromosome packed into a distinct region of the cell, which usually we call the nucleoid space. Okay. Many prokaryotes also have an additional small circular DNA called plasmids, and these replicate independently of the chromosome. We get to do all kinds of cool research with plasmids. They're just these little kind of extraneous circular pieces that kind of do their own thing. Really helpful for genetic research, but we're okay. All right, so some prokaryotes form specialized cells called endospores that remain dormant through harsh conditions. So this kind of gets us to that little bit of anthrax kind of question from the quiz. So you don't necessarily need to write this because I'm not going to test you over this, and neither will the state of Texas, but since we mentioned anthrax, it's worth talking about what this means. So endospores usually survive extreme heat and cold, and the endospore receives environmental cues that conditions have improved, and it'll absorb water, resume growth, and be awful. Okay, well, if it's, if it's anthrax. So it forms this kind of like dormant thing and just waiting for conditions to be right. Okay, this is a good place to take a break because we're going to switch gears here. So if you need to pause the video, take a break, do what you need to do, and then come back. So prokaryotes exhibit much more nutritional diversity than eukaryotes, allowing them to have it like everywhere. Okay, two of the sources of energy used. So we have phototrophs. I don't remember if we've covered this or not, but just in case. So again, photo is light, and then chemo is obviously chemicals. But the troph part of this talks about nourishment. Can't remember if we've covered this or not. Maybe we have. So phototrophs, they use light for nourishment. Chemotrophs use chemicals for nourishment. So notice sunlight produces to capture energy. Okay. And energy is stored in chemicals, so chemical nourishment for chemotrophs. There are two sources of carbon. So the last one was energy, and this is carbon. So if we're carbon, there are self-nourishers, which we're going to, nourish themselves with the carbon. Basically, think like the sugars. So when we're talking about carbon, we're kind of talking about those sugary things. So they can make the carbon themselves, or heterotrophs, and they have to get the carbon or the sugars from other organisms. So we have a whole mix of things, and these get really confusing, so I'm not going to spend too much time on it when we're limited already, but we do have like photoautotrophs, so they harness sunlight energy and use carbon, CO2 from carbon to make their own sugars. So it's use light to self-nourish. Yeah. Photoheterotrophs use light but different nourishments. So they get energy from sunlight but get their carbon atoms from other sources. It gets confusing. Chemoautotrophs, they get chemicals and they use those to self-nourish, so they harness energy from other chemicals and use CO2 to make molecules to make their sugars. Chemoheterotrophs use chemicals and acquire energy from different nourishments. So a lot of different things. Just to give you the idea, there are examples. So basically some plant-like ones called oscillatoria. Some interesting chemoautotrophs, which are sort of like rock-eating. We're not really sure what those are. Some that are photoheterotrophs. Rhodopseudomonas. There we go. I knew I'd get it in a second. And then salmonella is a chemoheterotroph, which is one that eats. Okay. Some other interesting things, so some connections real quick. Just know the basic definition, I'll give you the story here, but there's biofilms, so life films, that are highly organized colonies that attach to surfaces. Biofilms consist of one or several species of prokaryotes and may include some protists or some fungi. It's just, it's a good time. Lots of stuff are there. Biofilms can form on most any support, including rock, soil, organic material, or the surface of stagnant water. Biofilms communicate by chemical signals, they coordinate all kinds of things, and they'll collectively defend against invaders. It's a really nice community where everybody's communicating well, everybody's getting along, everybody's helping out. It's inspirational. Channels in biofilms allow for nutrients to reach the interior, waste to leave, and a variety of environments to develop within it. So it's pretty cool, and it sounds terrible, but how fascinating is it that in this particular tube where we've got this kind of nasty stuff, but it's all these bacterium and protists and various other things that are working together to help make sure that they all get nourished. So I think even down to the prokaryotic level, there's some of that whole nice guys finish first business. All right, prokaryotes are useful for cleaning up contaminants in the environment because prokaryotes have a great nutritional diversity, are really adaptable, and form these like super communal biofilms. We also have this thing called bioremediation, which would be nice to know the term. It's the use of organisms to remove pollutants from the soil, air, and water. So just again, we can use prokaryotes to do so much good. We kind of get over some that have given it a bad name. Fun facts too, again, I'm not going to highlight a whole lot here, but it's worth knowing that prokaryote decomposers are the whole reason that we have sewage treatment facilities. So raw sewage is passed through a series of screens. The matter that settles out forms a sludge. Gradually add a culture of anaerobic, so non-oxygen using prokaryotes, including bacteria and archaea, and the microbes decompose the organic matter into material that can be placed in a landfill or used as fertilizer. Yay! Yay, prokaryotes. Liquid wastes are treated separately from sludge. The liquid waste are sprayed on a thick bed of rocks. The biofilms, again, this nice community, grow on the rocks and removed all this dissolved organic material. Fluid drains from the rocks and it's sterilized, and then usually we put it out in the river or the ocean. Isn't that nice? So all this cool stuff that's happening here, we won't talk about it. Just bioremediation is really becoming useful as we kind of toxic up the waters and soil. Environmental workers change the natural environment to accelerate the activity of naturally occurring prokaryotes that would be capable of metabolizing pollutants. So we really can use them to our advantage. All right, look at them go. Okay, so to kind of bring it in with the evolutionary part of things, new studies of really the genomes of prokaryotes strongly support the three-domain view of life. So 3 domains means that we have one as eukarya, and then prokaryotes in 2 domain of bacteria and archaea. So they're separate, they're just different enough that we have 3 separate kingdoms. Okay, and we could go over it. Basically, it's just based on some RNA sequences, RNA polymerases, some introns, some, what is that, peptidoglycan is present, and a couple other things. All right. Just so we know a little bit about archaea, these guys are inhabitants of extreme environments. They're kind of extreme sports type guys. They're nuts. Okay, and archaea means old, so they're old and they just want to be left alone, and so they're in these extreme environments. Okay, they have some unusual proteins and other molecular adaptations that enable them to metabolize and reproduce effectively. So extreme halophiles just means that they thrive in really salty places, and we have extreme thermophiles, which live in hot water and acid pools. So phile means loving, halo is salt, so it's a salt-loving. Thermophiles, phile is still loving, thermo is heat, so heat-loving. All right, so there's some geysers for us. Let's see, we do have these other dudes called methanogens, so just in case, there's a molecule called methane. And it looks a little, it needs an N. It looks a little bit like this. It's just a carbon and it's 4 hydrogens. So you don't quite remember that from chemistry. That's methane. So there's some dudes that live off of methane, and we call them methanogens. So they live in these anaerobic environments. They give off methane as a waste product. Excuse me, I got it backwards. So they live in the digestive system of cattle and deer, decomposing materials in the landfill. If I don't remember to do it, you should look up it's a methane emissions of a cow versus a wallaby, and it's a cute little lecture by this old professor. All right, I forgot landfill. Okay, this is another great place to take a break and then come back, because we're going to go over 5 types of bacteria. So chunk your notes, write your summary, write your questions on the side, and come on back. So the domain of bacteria is currently divided into 5 groups, and those are the ones that we're going to look at. And that's comparisons of their genetic sequences. So we're going to talk about each of them, and we'll end up kind of making a chart so we know these dudes just a little bit better. So the first one is proteobacteria. These guys are all gram-negative, and they share a particular rRNA sequence, and they can represent all 4 modes of nutrition. So we're going to end up calling these guys gram-negative and diverse. Okay. So a lot to those, but those are proteobacteria. Theomargarita nabimbiensis is a type of photoautotrophic species of proteobacteria. It uses really sulfur to generate molecules from CO2, and it produces the sulfur waste that's this small greenish globules in the figure that we're going to look at. So all those little globules are this kind of like gross sulfuric waste. Okay. So we also have a rhizobium, a little easier to say. These guys are the ones that live symbiotically, so that means that they live, it just, symb is like together, bio is life, so it's just life together. So the things that are living life together are legumes, which are beans, and these rhizobium bacteria. So we call them symbionts. So they actually help convert nitrogen into a form that the legume can use. So it's a nitrogen fixer. All right. So again, symbiosis is a close association between an organisms of two or more species. So it just literally means life together. Okay. Endosymbiont, so endo means in, so we have life together in, which means one of them is within the other. All right. Rhizobium is an endosymbiont because it lives within the legume or the bean. Okay. Let me know if you need help clarifying that slide up. I realize I went quickly. Our second group are gram-positive bacteria. Gram-positive bacteria rival proteobacteria in the diversity, so also diverse. And they can include actinomycetes common in soil, which are going to be sort of fungi-like. We also have streptomyces, which are often cultured in pharmaceutical companies as a source of many antibiotics, including streptomycin. Okay, so those are some examples. This also means that because it's gram-positive, let's see if it's in the next slide. No. It also means that they're going to have a cell wall made of peptidoglycans. All right, there we go. That's the specifics there. Okay, there's some examples to look at. Cyanobacteria are the next one. Those are my favorites. If you're not sure of it, cyan is a color, and it's kind of like this bluish greenish color. It used to be in some old coloring stuff, but cyan is kind of a bluish greenish. So a bluish greenish kind of bacteria are going to be plant-like or photosynthetic. That's how I remember it. Look up cyan. I swear to God, it's color and it's pretty. Okay. So these guys are plant-like, they're oxygen generating, they use photosynthesis. And some species like Anabina have specialized cells that can fix nitrogen as well. So there's our little example there. Okay. And so you can kind of see them there as these are our little photosynthetic cells working together, and they have these kind of extra nitrogen fixing cells that can make nitrogen available to other species. Our fourth group is chlamydias. These live inside eukaryotic host cells. Chlamydia trachomatis is a common disease, cause of blindness in developing countries, and it causes non-gonococcal urethritis. Oh, Lord. Urethritis. Let's see. A lot of words, okay? The most common STD in the U.S., or I guess STI is sexually transmitted infection. Okay, so it is what it is, and all these guys tend to be parasitic and live inside eukaryotic host cells. Okay, so, but that is our example. Okay, we can see them within the host cells there. Spirochetes are the last one that we're going to look at, and these are our helical bacteria, so they're going to look spiral-shaped. These are notorious pathogens causing syphilis and Lyme disease. Do remind me to tell you about the Lyme disease story. But those are our two examples that are pretty well known. Okay, they look like that. So all organisms are almost constantly exposed to pathogenic bacteria. You probably have it around you right now. Don't freak out, but you do. All right. Most often, our body's defenses prevent those pathogens from affecting us. Namely, is your skin the way it is it's made to be kind of a drier environment that most bacteria aren't very successful in. You produce oils out of your skin. Oil also just doesn't go well with like living things. So oils, just the dryness, just the way it is, just isn't a good habitat for pathogenic bacteria. And it keeps it kind of away from us, so it's a good barrier. Most bacteria that cause illness do so by producing a poison. So exotoxins are proteins that bacterial cells secrete into their environment. So exo is exit. So it's a toxin that's leaving or exiting out of that particular thing. Then you don't need to know all this too much either. We're going to stick with just kind of the basics and knowing what those 5 types are, but at least we'll get exposure to a few other things. So endotoxins are components of the outer membrane of the gram-negative bacteria that are released when the cell dies or ingested by a defense cell. So it's a toxin that is within, so it has to be kind of killed for it to come out. All endotoxins induce the same general symptoms: fever, aches, and sometimes dangerous drop in blood pressure. Bacillus anthracis, which is anthrax, forms hardy endospores that have been used as biological weapons. So that's anthrax. All right. The weapon of C. botulinum is an exotoxin, so that's one that's produced outwardly, and it produces botulinum, which is the deadliest poison known. But here's the real kicker, is that botulinum blocks transmission of nerve signals that cause muscle contractions, resulting in paralysis of the muscles required for breathing. And so this is also responsible for the more benign use of botulinum for relaxing facial muscles that cause wrinkles. So when people say they're getting, oh my God, Botox, they are literally adding botulinum toxins to their face, which seems to be okay for a lot of people, but I don't know. I'll just rather take my wrinkles than putting botulinum in my face. That's just me, though. It is what it is. So how do scientists determine which bacteria cause disease? To test the hypothesis that a certain bacterium is the cause of a disease, a researcher must satisfy 4 conditions. These are called cost postulates. You don't necessarily have to write these down. It's just interesting, because I'm going to give you a quick story here, because I got 3 minutes before we hit the 30-minute mark, so we're going to do it. Okay, so find the candidate bacterium, in every case of the disease, isolate it, show that the culture bacterium causes the disease by putting it in a healthy subject, isolate that bacteria from the experimentally infected subject. That's what we do. So one of my favorite things of all time, which is like really a scientist taking this for the team, there was an Australian microbiologist, Barry Marshall, who hypothesized that chronic gastritis, also known as like a stomach ulcer, was caused by a bacterium called Helicobacter pylori, or H. pylori. Over the course of several years, Marshall satisfied the first 2 requirements, but his effort to infect animals failed to produce results. So at last, Marshall decided to take a radical course of action. He would experiment on himself. Honestly, this is not the first time this has happened. This story is great. He concocted a nasty brew of H. pylori and swallowed it out of the beaker like a champ. Right, seriously. Several days later, he became ill from gastritis, so step 3. He thought it was like, it's going to take a couple weeks. It's cool. Like, it's not that big of a deal, but it's like it took several days, and he was like, dear God, this is awful. And his stomach lining was teeming with H. pylori. He just had the stuff in there, and it was awful, and he really infected just the bejesus out of himself, which is the funniest thing to me. He did go do step 3 and 4, and he cleared up the infection with antibiotics. He continued to make progress in his research, and other scientists followed up with further studies. Several years after, his big gulp antibiotics became the standard treatment for ulcer patients, instead of like, just chill, don't stress out. It's like, no, it literally is a bacterium that causes it. So here's him, winning one of the Nobel Prizes for this research. Really took this one for the team. All right, so since his breakthrough research, scientists have learned that our relationship with H. pylori is ancient, at least 50,000 years, and it's complicated. Only a particular genetic strain causes ulcers. Others are harmless and just hang out. Some hypothesize that the absence of H. pylori can actually cause problems, but we're still investigating this kind of connection that's very complicated. Like if H. pylori in humans had a Facebook pages, their relationship would say it's complicated. Okay, that's the way that goes. All right, so we're almost there. I forgot what that had to do with it. I'm not going to worry about it. So with that, we'll start protists next time. We'll deal with that. We've got some stuff to worry about there. But there's a lot of information in that last bit, and we flew through some stuff. So if you're curious about what maybe you need to know or don't need to know, or what you see there, just send me an email, and we'll work our way through it.